Okay, we have protein synthesis and protein structure. When we talk about a protein, we talk about it as a protein. We don't talk about it as its individual parts and pieces. When we look at it from an individual parts and pieces, proteins are comprised of a number of various different amino acids. Okay? And so it always helps to kind of visualize a protein as just a string of beads. Okay, and that's what we got right here. We have a string of beads. Okay? And we have 20 different ones. And we just looked at those. We have essential and not essential. And they all get put together in a specific sequence, and they can help to give the blueprints, right, to make certain structures throughout the body. Ultimately, it's DNA that puts these sequences of amino acids together, right? What's DNA? Deoxyribonucleic acid, right? It's your hereditary information. We have genes that code for the individual proteins. The dark hair, certain gene, certain number of genes code for it. Height, makes collagen, makes bone structures. All various genes, all making very specific things. Okay? And we can assemble these proteins or these amino acids to make specific proteins according to that fantastic information within that DNA. And when we look at it, the book gets into it. You know, I'm not going to ask a short answer question based on it because it's more gen bio type questions or upper level biology questions, biochem, if we really want to do a deep dive into it. But we have two processes here that involves the reading, copying of DNA, and the reading of DNA to make a protein. Those are called transcription and translation. With transcription, we are making a copy. We're just transcribing, right? We're transcribing the genetic code. So this is happening inside the nucleus, where we read the DNA and copy the instructions down. Then that DNA is moved from inside the nucleus out into the cytoplasm. If you've had gen bio, you know it moves to the nuclear pore as messenger RNA. Out in the cytoplasm, this is where I read it. And when you read something, you are translating it. So out in the cytoplasm of a cell, we read the copy and we make the protein. Okay? So transcription is just copying down the instructions from the DNA, forming messenger RNA in the process, heads out into the cytoplasm where we translate it, where we read it and make the protein. And if we were to look at this, this is what it looks like. All right. Of course, I'm skipping out a whole vast number of steps inside with transcription. Out here with translation, all the various different processes that cause that to happen. But again, that's gen bio. I'm not going to get into that. What I want you to know is what DNA is. It's the genetic or hereditary material. The gene is the portion of the DNA that codes for the specific type of protein, the specific sequence of amino acids. And this process of copying the DNA and reading the DNA ultimately involves two processes, transcription and translation. One, we make a copy of the DNA happening in the nucleus. One, translation occurring out in the cytoplasm where we are making the protein. And again, that's what this is trying to show off here. Granted, with a lot more steps, but we don't need to do that, okay? Makes a great multiple choice, a vast number of multiple choice types of questions. When we look at this string of amino acids, if I go back here, here's our string of amino acids making a protein. You can see there is a bond that connects these two. Sorry, guys. There's a bond that connects all of these amino acids together. This bond has a specific name. This bond is called a peptide bond. Okay? So if I was to ask you what connects two amino acids together, you would say a peptide bond. When we start to string these together, we could have a dipeptide, tripeptides, polypeptides, two amino acids strung together. Tripeptide, 3 amino acids strung together. Anything greater than that, we're going to have a whole lot. And those are polypeptides. Poly, again, means many. Okay. And these can be upwards of 50 or even a hell of a lot more different amino acids strung together. And when we think of proteins, the most naturally occurring ones, everybody, are polypeptides. All right. When would a monopeptide or dipeptide or tripeptide exist? Monopeptide, that don't exist. Dipeptide, tripeptide exist. Those exist when we're going to be talking about the digestive process. We break these amino acids down into individual amino acids in the digestive tract. When we look at a protein structure, it has 4 different levels of complexity. The most basic, the most simplest form is the primary structure. Okay? All it is just the string of amino acids. Okay? Linear chain of amino acids linked by peptide bonds, yada, yada, yada. Okay? This is ultimately the various different amino acids that form the ultimate structure, whatever it's ultimately going to become. Bone, collagen, whatever, elastic fiber, whatever. As these start to get long, as this primary structure starts to get longer and longer and longer, it'll start to coil on itself. When it starts coiling on itself, it forms a secondary structure. As there's more and more amino acids there, we start to get much more interaction between all of the various different types of amino acids. They all have various different properties. And this coiling phenomena arises from the hydrogen bonds. H is hydrogen. Okay, H is hydrogen. And this hydrogen bond is going to form between all of these kind of adjacent, localized amino acid. Okay, and it gives you that coiling structure. We see the same hydrogen bonds with DNA. DNA is a deoxyribose nucleic acid, 2 strings of DNA twisted around each other. It's a double helix, right? And that double helix, each side of it is held together by hydrogen bonds. Same thing here. Again, we can have hundreds of these things strung together here. As those continue to grow in length, excuse me, it'll take on another layer of complexity and become a tertiary structure. With this, we get into a lot of chemistry and a lot of various fantastical interactions with them. And these depend on the chemical properties that are the various different types of amino acids. Some have polarity, some are polar, some are nonpolar, some are acidic, some are basic. It all depends on this R group. Remember that R group is the functional group. Okay, this is the shorthand for function. And I'm just going to put functional. Okay, remember that's the functional group. It's that R group that gives these amino acids their true nature, which is polarity, non-polarity, acidic, alkaline, or basic. Okay, and this helps to give it this fantastic 3D, very twisted, very globular type of structure. For the most part, this is where proteins stop. Right? Not everybody goes to the next level, the quaternary structure. The quaternary structure, this is where we have multiple tertiary structures bound together. As you can kind of see, structure of protein that's comprised of 2 or more polypeptide chains arranged together in a unique manner. It's a bunch of tertiary structures strung together. Okay, and you can see they're all color-coded for your pleasure here. We have 1, 2, 3, we have 4 different structures here associated with this quaternary structure. And again, not everybody gets to this level. Okay, not every protein goes to the 4th level of complexity. Most of them stay at that 3rd level. An example of this is hemoglobin, myoglobin, hemoglobin. Okay, they're both examples, but hemoglobin is the one I have listed here. So if I was to ask you an example of one on an exam as a multiple choice type question, I imagine you'll remember that. Okay, very complex in structure. Hemoglobin, super complex in structure. Come to phys or come to anatomy and phys class, we go over it there a little bit more in depth. But these are the levels: primary, secondary, tertiary, quaternary, basic, most complex. Not everybody gets to this level. Okay? Most of them stop right here in complexity. And then we chew it up, and then we digest it to try to get it down to this primary sequence so I can cleave off all of those during the digestive process. The shape of a protein is important. This sequence of amino acids is vastly important. Just changing one amino acid can change the entire functionality of the structure. And no better example than we see with right here. One little amino acid is not included in the proper sequence, and we get a total different shape and structure for the red blood cell. Sickle cell anemia is an inherited type of anemia. Anemia means low red blood cells. And sickle cell anemia is characterized, as you can kind of see here, by this sickle-shaped red blood cell structure. And again, just one amino acid has been lost. There's some kind of misread, and we've not included one specific type of amino acid, and we get a total different structure. Of course, this is an advantage in certain situations. Sickle cell anemia, the shape of it prevents the plasmodium, the bad guy behind causing malaria. This shape actually prevents malaria from getting in there, from succumbing to malaria. Okay? Prevents the plasmodium from getting inside the red blood cell and dividing. So sickle cell anemia does have a reason to be there. It's just not one of those totally negative things. Now, if you don't live in areas that are endemic to the Anopheles mosquito that carries malaria, then it's not really doing anything for you. Those of African American descent, those of Eastern European Jewish descent, those are the two ethnic groups that generally have the gene that carries for the sickle cell trait. So, but it is important, okay? Reading that DNA is important, okay? And there's a lot of DNA that has to be read in the right sequence to make the right stuff. Ultimately, we want to break down proteins, okay? We want to break down that tertiary structure into a way that we can open it up so we can break up those peptide bonds, all right? And that's just referred to as protein denaturation. When you denature a protein, you alter the shape of the protein. Again, when we alter the shape of the protein, we alter the functionality of that protein as well. The great thing about protein denaturation, it's okay. The cooking process actually denatures proteins and makes it a little bit easier for our bodies to do what they do, to break down those proteins to their individual amino acids. So we can digest them and we can absorb them. Okay? It can be heat. It can be alcohol. We can physically agitate or whip it up. That's enough to destroy the structure of this thing, so it makes the enzymatic activity mean something. If it's in that complex tertiary structure that you see up there at the very top, very difficult for enzymatic activity to get in there and to be really effective in doing what it do. So, eating food that's cooked is helpful and beneficial. Can you eat stuff raw? Sure. Is it a little bit more difficult to digest and break down? Heck yeah. Heck yeah. And you've seen this. You've ever cooked eggs on a skillet before, on the stovetop? All that egg albumin that you see towards the edge, that gets really, really, really hot, gets broken down. And it's not edible, is it? So protein DNA trace, this is part of the normal cooking process, and it's the digestive process, acids of the stomach, right? That's where protein digestion occurs. I'll break that protein down and open it up so I can have my enzymatic activity mean something.